Our next panel for the day would be on COVID-19 test kits, the future prospects. It's a very important panel discussion with a lineup of very good experts we have in. We have Dr. Rohit Chawla, the Associate Professor, Department of Microbiology, Maulana Azad Medical College and Associated Hospital, New Delhi, as our moderator. Thanks a lot, Dr. Rohit, for accepting our invitation and also moderating the session. And we have Dr. Saurabh Kumar Banerjee, Dean and Associate Professor, School of Pharmaceutical Management, the IIHMR. Thanks a lot, Dr. Saurabh, for coming in to share your views. We have Dr. Shivaji K. Jadhav, the head molecular biology COVID laboratory SRL for this hospital. Thanks a lot, Dr. Shivaji, for coming in. We have Dr. V. Vasuki, principal investigator, VRDL, Department of Microbiology, Government of Tiruvallur Medical College. Thanks a lot for joining in, ma'am. And we will be shortly joined by Dr. Shanta Datta, Director and Scientist, GICMR, National Institute of Cholera and Enteric Diseases. So it's a very pertinent, panel with very important subject to deal with. So I would like to give the dais to Dr. Rohit. Please carry on, sir, with the discussion.
At the outset, I'd like to welcome the panelists and the audience to this panel discussion on COVID-19 test kits, future prospects. As you are well aware, until mass vaccination for COVID-19 is achieved, screening, early diagnosis, continuous surveillance, and epidemiological contact tracing would remain critical strategies for containing the spread of this disease and mitigating its profound impact on global health and economy. COVID-19 pandemic in many ways will remain a watershed moment. Never before in the history of human civilization have the containment strategies for a pandemic been driven by such a rapid and extensive deployment of diagnostics. To put things in perspective, on 8th of December 2019, the first case of COVID-19 was identified, and by 10th of January 2020, the first genome sequence for SARS-CoV-2 became available. So just tells you how fast-paced the diagnostics were with regard to identifying the causative agent and also sequencing it. This quickly led to the rapid development of the nucleic acid amplification assays for the diagnosis of COVID-19, which was followed by the development of various antigen and antibody-based assays. This panel discussion attempts to unravel the landscape of bioanalytical testing for COVID-19 with a focus on the performance metrics as well as the limitations of various techniques. The panelists would initially discuss about the state-of-the-art technologies that are limited to centralized laboratories, which often set the clinical metrics against which the emerging technologies are evaluated. Through this panel discussion, we also hope to touch upon rapidly emerging point-of-care technologies which open the route for testing the community at home at points of entry to widen the screening of individuals. No doubt, the new technologies developed in the context of COVID-19 will help advance the diagnostic field for the immediate use, that is dealing with COVID-19 pandemic, but more importantly, it would help towards building preparedness for the future infectious disease pandemics. It's not a question of if the next pandemic would occur, it is a question of when. So I believe that the investment which has been made in the development of diagnostics for COVID-19 would bear fruition in dealing with the future pandemics when they occur, and not if they occur. So without further ado, I would lay out the format for today's panel discussion. Dr. Vasuki will dwell on RT-PCR-based assays, which will be followed by discussion on isothermal amplification technologies, including CRISPR by Dr. Jadhav. Subsequently, viral antigen detection assays would be covered by Dr. Shanta, while a last speaker, Dr. Saurabh Banerjee, would touch upon assays for COVID antibody detection. I would humbly request all the panelists to kindly stick to the allotted time, that is 10 minutes each. The panelists would address the questions from the audience once all the presentations are completed. The audience are encouraged to submit the questions through the QA box, which is there, I think, towards the left of your screen. So without further ado, I would now hand over to Dr. Vasuki. Over to you, madam, for your presentation.
I just sharing my screen. So I am going to talk about RT-PCR based assays. This is the gold standard for COVID-19 diagnosis. Here we are detecting the part of viral genome, that is RNA, in the respiratory tract specimens like nasopharyngeal swab, oropharyngeal swab, lower respiratory tract specimen like sputum, and lower respiratory tract aspirate, BAL, bronchoalveolar lavage fluid. We are going to detect the part of viral genome. The target genes used to buy various commercial kit are varying according to the manufacturer. The things are E gene, envelope gene, M gene, N gene, nucleocapsid gene, spike gene, and non-structural genes like RDRP, RNA-dependent RNA polymerase, then like open reading frame, ORF1A and 1B. These are the target genes we are detecting in the patient specimens. Till now, the RT-PCR are having high clinical sensitivity. It means low false positive results occurred. In fact, in some settings, we are facing we are encountered false positive, false negative results. Those because of inappropriate sampling technique and storage, transportation, and purification process, RNA purification process, they will result in false negative report. Sometimes false positive also we have encountered like pipetting errors, cross contamination during processing of specimen will result in false positive result also. This is the only mode of diagnosis for early diagnosis of the COVID-19 patients. Even before onset of symptoms itself, we can detect the disease by RT-PCR diagnosis. And we can perform large number of tests consistently. This is the one way we can detect large number of tests can be done in a day. So mass screening is possible for a pandemic like this we faced last year and now also. Then quantity reduction of SARS-CoV-2 RNA also possible because we can correlate the CT value with the viral load. Even though we are having some challenges in the RT-PCR test, like it's a very complex test and expensive. So the setting up of lab with the biosafety level 2 is very expensive. Even though our government supported a lot, spent a lot of money regarding for this expensive test. Then slower to deliver the result. It means the turnaround time is it will take more than 24 hours, even though the analysis time is 4 to 6 hours inside the lab. From the sample collection to deliver the report, it takes more than 24 hours. So these are the challenges we faced from the starting time till now we are facing the challenges. Coming to the point of care testing, here the available test in India are CBNet, cartridge-based nucleic acid amplification test, including Gene Expert and TrueNat. Here the rapid result we can take. Here the integration of the nucleic acid extraction with the amplification and reduction on a microfluid chip is available. It will reduce the reduction complexity in the diagnosis. So unrestricted test site is enough. So no need for a biosafety cabinet tool is not needed. So it's a point of care testing. We can use in the periphery setup also we can use. Low professional skill requirement for the operators is enough for conducting these tests, point of care test. The one more modification in the RT-PCR, that is the newer version is Dry swab extraction free RT-PCR. This is the modified form of RT-PCR introduced by the CBM, that is the CCMB, Center for Cellular and Molecular Biology. Under CSR, they introduced the technology. This is a modified version of the RT-PCR, that is a dry swab extraction free RT-PCR method. Here we are using without VTM, we are collecting the sample and it will be treated with the buffer solution and incubation. No need for RNA extraction is not done. So when in settings without automated RNA extraction, we can go for this type of test, dry swab method, RT-PCR, extraction-free RT-PCR method. We can. This is the modified version introduced by our government. So in many setup, we are having the automated nucleic acid extraction equipment. This is the another advantage. In future, efficient and fully automated nucleic acid extraction equipment will provide definitely better production in terms of detection sensitivity. That is, false positivity will be lowered and definitely personal safety will be there. So in future, the molecular diagnosis research for COVID-19 infection will speed up the sample preparation. Everything will be automated. Sample preparation, increase detection throughput and accuracy. It will improve the detection automation level. Currently, we are having automated level at the nucleic acid extraction only, but the detection level we should improve the automation level and develop the novel technology with low requirement and low cost for equipment and testing personnel. Because manpower is very important, the training of the manpower, trained manpower is shortage all over India. So we should improve our technique according to this requirement. So another crown for this diagnostic is molecular diagnostic for COVID-19 is digital PCR. This is the third generation PCR. So first generation, in compare with our RT-PCR, it is used for absolute quantification of the nucleic acid molecules. It can directly detect the copy number of the target sequence and detect the limit. Detection limit can reach a single copy. Here the important difference here is initial sample mix is partitioned into large number of individual wells prior to the amplification step. So it will result in either one or zero target in each will be there. So absolute quantification of the nucleic acid molecule is possible with this digital PCR method. So sensitivity, specificity, accuracy, resolution, tolerance is very high in digital PCR. Digital PCR is a powerful method. The advantage of which include improved CNV detection. CNV means copy number variation detection and accurate NGS library quantification, like next generation sequence quantification is possible with this method, and increased sensitivity for detecting the target in limited clinical sample. This is very important. So we can detect even a single copy of RNA can be detected with this digital PCR method. And improved multiplexing capability. Many number of targets can be identified by multiplying the multiplexing the machine with digital PCR. We hope in future the molecular diagnostic research will be improved and we will meet all the requirements in future. Thank you, sir.
Thank you, madam. We'll take questions at the end of all the presentations, as was mentioned earlier, and we'll carry forward the discussions after all the presentations are over. Now, I would request Dr. Jadhav to shed light on the isothermal amplification techniques. Essentially, PCR is a technique wherein we use cycling of the temperature parameters to be able to achieve the target amplification. However, we have now new technologies coming in where you don't require the cycling of temperature conditions and you can do it at the same temperature. So to shed more light on these techniques, what are the advantages, disadvantages, what is the current status, I would now invite Dr. Jadhav to give his presentation. Over to you, Dr. Jadhav.
Thank you, Dr. Rohit Chawla, for the warm welcome. And I'll start my talk on isothermal amplification. On the offset, I'd like to thank the organizers who given me this opportunity to give a talk on the important topic of COVID, especially this in the molecular diagnostics, RT-PCR, and other novel technologies which are coming up. It's very important talk where I wanted to elaborate more on the different techniques. So I wanted to open up my screen.
There's share button at the bottom.
Yeah. Yes.
Yeah, it is coming on.
Yes. So I welcome all the panelists who are involved in the important topic of COVID-19 testing kits and future prospects. So my topic will be covering on important aspect of isothermal amplification assays by using different technologies. So as you know, SARS-CoV genome, it's a 29 kb genome which has a different structural protein viral components. So when you look at the detection in the molecular level, so you can see the RNA. So laboratory-based tests are mainly focused on the components based on the RNA. These are the molecular diagnostics, and the different strategies can be used. So in detection of RNA, there is the RT-PCR based on isothermal, LAMP assays, NGS, CRISPR technology. These all are based on the RNA-based assays. So some of the protein-based assays also can be done, like antigen, ELISA, antibody. These will be covered by my next panelist, I think. So I'll give more focus on the isothermal amplification, LAMP assays, and the CRISPR technologies. So when you compare the nasopharyngeal swab, so the RNA-based technology is divided into different components. So you can isolate the RNA, viral RNA, and further proceed for RT-PCR as well as isothermal techniques. And based on the viral antigen, so you can go for antibody assays, ELISA, immunoblot, and different assays. So for this serological and nucleic acid based, there are two categories. So serological based you can do with the directly with the blood sample. You can go ahead with the antibodies and go on. So mine is mainly core into nucleic acid based, where it is highly sensitive, highly accurate. So there will be chance of getting a viral antigen false negative as well as false positives. But when you go with the genome molecular like RNA component level, it is very, very accurate and sensitive. So as of now, the gold standard is RT-PCR. So RT-PCR, anyway, Dr. Wasuki has already explained about the advantages and disadvantages. So I'll not go detail about the RT-PCR advantage, disadvantages, but I'll go with the advancement in the different technologies by using isothermal reactions, NASBA technique, and CRISPR techniques. So when you look at overall screening, so the sample collection is proceeded with this technique. So it's the well trainers like phlebotomists, they'll collect the sample accurately, and they'll be transported in a special transport medium. And there are different techniques we can use it. So once the sample is collected in the VTM, they'll go for the lysis steps and extraction steps. And further, you can divide your technologies. So you can go for RT-PCR, you can go for isothermal LAMP amplification. So you can go for further based on your CRISPR detection. So these all are the different platforms. Once you have your RNA, so the main basic component is RNA. From that, you can disaggregate your technologies. So based on your expertise, your utilization, your lab manpower, your equipment, laboratory facility, based on that, you can divide your things. So as you consider, isothermal amplification is a very simple technique where you can utilize a different set of primers and probes. So I wanted to give detail about the isothermal amplification. I'll go about the NASBA technology and CRISPR technology, where I don't want to focus more on antibody-based assays or sequencing by Sanger or NGS or whole genome sequencing of the virus. So these are the other techniques which can be utilized. So this will be useful in the large geographical studies where you can go for the viral genome, viral distribution, and studies based on epidemiological surveys. You can do a larger preparation, or you wanted to identify any kind of variants, you can go with the sequencing based assays, Ithorpor, Nanopore, Ithorpor, Illumina, different platforms you can use for sequencing. So as RT-PCR already discussed, it has a limit of detection. So SARS-CoV RT-PCR detection limit is less than 10 copies, you cannot detect it. So the amplification, sometimes it may need false positive or false negative because of some of the cross reactivity which occurs between the primers when you use for amplification. And also the amplification depends on the expertise, how he handles the samples, process it till you get the accuracy of the results. It all depends on the expert manpower. It is required and expensive instruments, and you need a skillful manpower to analyze the CT values, analyze the different temperature, any variations, any reproducibility, any presumptive positive. So it requires a lot of expertise. So based on that, they think of going for the alternate technology, that is isothermal reaction. So RT-LAMP is a method which shows effectively it will detect the samples of COVID-19. It has been reported by Yan et al. In 2020. There is one of the publication where you can see the RT-LAMP is applied for COVID-19. So it involves multiple loop of primers, which includes ORF, NAB, S-gene, and this is a technique which involves a strand replacement activity, and it is very, very targeted. So you can detect up to less than 20 copies per reaction also, and it will be very sensitive. These results are very, very compatible with the RT-PCR, as they are giving sensitivity up to 100% and very accurate. So it will be detected by different technologies in less than 30 minutes also. So this is the overview how the lab will be worked. So it's a point of care testing. We can use the lab. It has a different method. So you have a heat inactivation. Water bottles can be used. There is no need of expensive instruments. So the RT-LAMP is again the reverse transcription step occurs. Then there is a different set of primers you have to use for amplifying the complementary cDNA. And there is a formation of BIP. So this BIP will be detected by different methods. So recently Abbott has got a point of care testing by using isothermal amplification, and it had a very shorted turnaround time of 30 minutes. It has been approved by FDA also. So this is a very simple technique where you have a biological sample collected and the DNA or RNA is extracted. So you will be mixed with the LAMP master mixer, which includes all your primers and your detection dyes. So it will be lyophilized things, and then when you have a color detection, you will observe the negative one will not have any change in color. The positive have a change in color. So it all depends on your streptavidin codeine binding affinities based on affinity. So there are different methods also. You can go with a lateral flow assay by using the strips also. You can detect that by a calorimetric fluorescent dye change also. And you can also go with the gel-based detection also. It's all you have a different technology to visualize the amplified product or detected RNA of COVID-19. So the difference, if you compare the RT-LAMP, so RT-LAMP is isothermal mediated, whereas the PCR stands for repeated reamplification. In PCR, you have different steps, 3 steps: denaturation, annealing, and primer extensions. And in RT, you have only one step process. You will not need to go for repeated different cycling conditions where you need a sophisticated instrument like RT-PCR. So it requires manpower, extensive training. The amplification is not occurring in PCR. It will occur in a different temperature, where in the RT-LAMP it occurs at the 65 degrees centigrade. There is a constant temperature is going on, and the temperature fluctuation and all those, it will not hamper in this reaction, because it is very easy and very simple methodology where you will have detected by different fluorogenic components. So when you compare the real time versus the ramp, the real time LAMP is highly sensitive and it will be done in 1 hour. When you compare the RT-PCR, it will take minimum 3 to 4 hours, as you have to do the extraction till different cycling conditions, till the analysis. It will take 3 to 4 hours. And when you go with the instrument level, there is nothing is required for RT-LAMP. Where you have some stalbidometry is fine, or you have a heating block is enough for that RT-LAMP. Where in RT-PCR you require a sophisticated instrument like RT-PCR machine. So the user-friendly in RT-LAMP, there is no carcinogenic material is available. When you go for some kind of gel-based detection, it will have a carcinogen like ATBR. So these are the comparison between the RT-LAMP and RT-PCR. So NASPA is also one of the techniques which is also based on the isothermal amplification of the primer binding, where it uses again the strand displacement activity, and it will be annealed with the targeted sequence of the viral genome. So the amplified product will be again detected by different techniques like photometry, and this will also be developed with the fastest available point of care testing, where the SARS-CoV can be detected. So this is one of the novel technique where recently has come up with a collaboration with CSIR IGIB. It's called CRISPR-based diagnostics. It is named as a Faluda test. So this is very affordable, and it's like a pregnancy-based test kit. So it has used a CRISPR-Cas technology where you have a better sensitivity and specificity. The specificity will be achieved by using a Cas13 technology, where it has a very selective binding of the viral genome. That's why it will achieve more sensitive. And it also has a collateral cleavage also, where you can identify the specific targeted sequences. So it is very affordable, cost effective, and very cheaper also compared to the other technologies. And this is developed by Tata Medical Diagnostics by using a Tata MD check. And these are the strip based we can usually visualize whether it's positive or negative. The only thing is in this we can see is the extraction step, where the identification will be done in less than 30 minutes, 30 to 45 minutes, but extraction will require a time. So if it is a manual extraction, it will require at least for 8 to 10 sample, you require at least half an hour. So that if you add the extraction as well as identification, it will come around minimum of 75 minutes. So it is a very quicker and rapid technology and very sensitive. So if you want to go step by step of this also, it's again like a person has to give a sample from nose or throat, osopharyngeal or naropharyngeal. The RNA can be isolated quickly either by manual method or by automated extractor. The RNA can be put in the PCR where you can amplify it. Then the amplified product can be added with the CRISPR-Cas9 technology with its affinity to bind. And the next 2 minutes, the positive strip will show the positivity or negativity based on the binding affinity. So the amplified product will bind to the specific CRISPR RNAs, and it has a sensitivity of around 96% and specificity of around 98%. So it will take very rapid. It will be detected in less than 1 hour. So this is a very advantage we are having. So I have compiled overall methods and their key features. So I'll not go with the RT-PCR where it requires more than 2 to 5 hours of time. So I'll focus only on the RT-LAMP, where you can take out the sample from oropharyngeal, nasopharyngeal, go for directly RT-LAMP, where you will get a result less than 30 minutes. So accuracy is to be determined. It has very specific, very sensitive. Sometimes it requires some kind of validation before you go for a LAMP assays. So that is the one drawback. And the CRISPR is already, everyone is a very cost-effective, rapid diagnosis. Only is a extraction procedure. So if it is automated, it will be very faster compared to the other technologies. And most of the technologies, which includes RT-PCR or LAMP or CRISPR, so all are giving highly specific. These all are molecular based on the RNA. So it will be very specific and very sensitive. So I would like to conclude based on all the markers, detections. So these not only one technology, the many of the techniques all depends on how the accuracy. This also depends on not only on the accuracy of the person who's handling, the person who is performing the experiment. It also depends on the sample transportation, how the sample is collected, how the sample is transported to the lab, which is the transport medium you are using, which is the temperature condition you have brought to the lab. So everything depends on accuracy and sensitivity based on these not lab clinical parameters. So other parameters also plays an important role in accuracy of diagnosing the COVID-19. So I'd like to thank you very much.
Thank you, Dr. Jadhav, for a comprehensive presentation on the isothermal techniques that are available. So just to put it in perspective, there are a lot of technologies which are coming up, and some of these have already been approved by ICMR, particularly RT-LAMP that was referred to by Dr. Jadhav. The beauty of this technique is that you don't require a costly equipment to carry out the amplification process. The same can be carried out at the same single temperature using equipment which are not very expensive. The only thing is that the extraction part remains the same, as is the case with the conventional real-time qPCR. In addition, it was mentioned by Dr. Jadhav that other techniques for detection like CRISPR-Cas have become available. One of the tests has also been approved recently by ICMR, the Feluda test, which is named after a famous detective in series in Bengal. The CRISPR-Cas assay makes use of the gene editing technology. The initial part, that is the extraction, and thereafter the amplification again remains the same. But the good point is that the old PCR machines can be used We don't require a real-time PCR machine for amplification of the target for CRISPR-Cas system. And once you amplify the target, it's only the detection which is being carried out by using the CRISPR system. The beauty of the CRISPR system is that it can be put into a paper format, which is the case with the Feluda test, which is available right now. And you can have a rapid visual detection of the strip, and you can get to know whether the result is positive or negative just by putting up simply a paper-based test. It can also be modified into a fluorescence-based test, though such kind of a test is not currently available in India, but then it is envisaged that it will become available in times to come, where the fluorescence-based detection is there, just like what we have for RT-qPCR. So thank you very much again, Dr. Jadhav, for shedding light on the isothermal technologies and where they stand in the overall landscape of COVID-19 diagnostics. Now I'd request our third speaker, Dr. Shanta Datta, to shed light on another totally different diagnostic technology, that is the antigen-based assays for COVID-19. Over to you, madam.
So thank you very much, Dr. Chawla. And I think this is the right time when this is being discussed about all the tests available for detection of COVID-19 infection. So we have heard the first two panelists already have discussed about the molecular assay as well as the LAMP-based assay. So I would like to discuss on rapid antigen test, which are available for detection of COVID-19 infection. So as we all know, because this is a test which depends on antigen detection, so it is naturally one antigen antibody reaction. And this antigen is actually the foreign molecular structures that can trigger immune response in our body. And antibody is also a protein, but this protein is evoked or generated in response by the immune system of our body when our body recognizes one target antigen in some viral or bacterial pathogens. And this immune system, immune response is meant for destruction of the antigen. Now, these are different types of tests available for COVID-19 detection. So I will be talking on antigen tests, where the target is detection of viral proteins. There are several proteins in this virus. The most of the antigen tests are based on detection of spike protein, which are present on the surface of this SARS-CoV-2 virus. The underlying principle is that already in the test kit, the antibody is coated, and that antibody, if the antigen is present in patient's sample, clinical sample, then it will capture the antigen on the test strip, and it will generate some color which will be visible in naked eye. And it is a point of care test, and the test results also come very quickly within 15 to 20 minutes. So what are the key facts for rapid antigen test? This test can contribute for overall diagnosis of COVID-19 testing. The main advantage is the turnaround time, like we can get the result within 15 to 20 minutes. But the major disadvantage again, this test is less sensitive if we compare it with uh, RT-PCR or LAM-based test. So in that respect, one has to be careful that it might give false negative result for a actually positive patient. So it usually gives best, it performs best in case of high viral load of when the patient suffers from early symptoms or in symptomatic level. Within 5 days of onset of symptoms, it gives positive. But and the minimum performance requirement as said by WHO should be more than 80% sensitive and more than 97% specific. Then only this rapid antigen test should come to the market. But there are some kits are available commercially, but all the kits, they do not fulfill this criteria that more than 80% sensitive and more than 97% specific. These tests are specific, but sensitivity is quite low. They may be sensitive as low as 50% sensitive. Tests are also has been observed. Then country needs to perform independent. So it is very important that site specific, setting specific or site specific validation of each and every rapid antigen test should be done before it is implemented for use. So where we can use the rapid antigen test and how it will be beneficial, usually I have already spoken that it is appropriate to use in high prevalence setting where the number of the infection or the cases are high. But in low prevalence setting, it might give positive for highly infectious cases, but again can give false negative if the prevalence is too low. Then rapid, but in one way it is beneficial, because if the highly infectious cases can be detected by rapid antigen test, then it helps reducing further transmission. And this is because it takes only 15 to 20 minutes time for giving the results. So early detection of high infectious cases, which is it can detect early, it can make early detection of high infectious cases, which are very important for reducing the transmission of the virus. Then so it has got some public health objectives, like it can detect very rapidly, so which is very important for prompt clinical management, and which is again important to reduce the mortality of the disease. Then the transmission may be controlled if early diagnosis followed by isolation can be done. So it is important in specifically in healthcare settings and social care settings by early detection and isolation of the cases. So this is just, this gives the diagram how we will go for if antigen rapid diagnostic tests are used. So this is the preparation of sample collections. This we all know. One should have the proper protective equipments, and then with swab, you have to take oropharyngeal swab or nasopharyngeal swab. But and there is along with the kit, one for squeezing the swab or rubbing the swab, one buffer is provided along with the kit. So this antigen for antigen testing, these are the preparation. It doesn't need any sophisticated equipment. It doesn't need any skilled personnel. It can be done at the bedside, even at the community level. So only these few things are required. And the test kit, then one timer that after result should be taken after 15 to 20 minutes. Then extraction buffer tube, it comes along with the kit. The medical waste container, because after the test is done or it is performed on the kit, then it has to be discarded because it has the samples, and so it is infectious, so it has to be discarded with proper medical waste, following proper medical waste standard medical waste rules and regulations. And disinfectant is also important. The bleach and alcohol can be used. so that the transmission can be contained. Now, after proper collection of the samples properly, then this is the second panel. It gives how the testing is done. Insert the swab into the extraction buffer tube and then squeezing the buffer tube and starting the swab so that the entire virus or the antigen what is present in the sample swab that can be transferred to the buffer. Then press the nozzle cap and tightly on the tube, and then just apply a drop, 1 or 2 drops as it is given in the insert of the kit, and allow to wait for 15 to 20 minutes, and then the kit or the test result needs to be read and interpretation has to be made. Then dispose the gloves, test device, swab and extraction buffer all in a container with having some bleach or alcohol so that it may be inactivated, the antigen may be inactivated. Now coming to the interpretation of the test result, we must keep in mind that if the target antigen is present in sufficient concentration, then only it gives positive. So I have already told that the only disadvantage with this is that it is less sensitive. So, and it will bind to specific antibodies fixed to the paper strip. And the strip comes in plastic casing. Then there will be a detectable signal which we can interpret after 30 minutes. The 2 bands usually appear. One is at the C and T, C for control and T for test. So if both the bands come, then it says that the sample is positive. Here there is a mistake, like if the test becomes negative and the only band comes in the control, that means the test is valid but the sample is negative. And if no band comes in either C or T position, then it says that the test is invalid because the control band has not come, so which makes the test invalid. So ICMR has also recommended deployment of these rapid antigen tests in the following settings. When although the number of the cases has gone down, but there was in the past, we have come across some time when in India there were more than 1 lakh positive cases per day. So during that time, it was not possible to get the all testing done by RT-PCR, which needs at least, we have heard from our earlier panelists that at least 24 hours required to get the result. So in those setup, this is the ideal test which can be used. So in containment zones at the community setup, it can be used by the state government or the government. Actually, in our country, all the government workers or the government personals, they have been used for using this test. So this has been used in central or state government medical colleges, hospitals, NABH accredited hospital healthcare, as well as in all private labs which are accredited by NABL and also recognized by ICMR for COVID-19 testing. So FDA has authorized this rapid antigen test in December 15. It is a milestone in diagnostic testing for COVID-19, because by authorizing the test, it can be used not only by the government personnel, but it can be used even by the patient himself. It can be used for over the counter, and it can be sold over the counter in the drug stores, and the test result can be obtained within 20 minutes. Here one term is used, that surge testing. This is surge testing is the rollout of additional community testing by the people who do not have any coronavirus symptoms. So it is being used by the scientists as well as public health officials to know the mutants of the COVID-19 virus. Like, because there are several mutants have been identified already and published also, but these all mutants does not cause a see change in the antigen. So antigen will be detected even if it is not detected by the sequencing or RT-PCR. So antigen can be detected by this rapid antigen point of care test. So this is one advantage by which we can identify if there are mutants present, and also I already spoke that it helps containment of the spread of the infection. So these are the following conditions where it can be used. No, if if any hospital or any testing facility wants to use this POC antigen test, then they need to register with ICMR to obtain the login credentials, and then initiate the testing. And all the symptomatic negative patients, if it comes negative with POC test, it should be referred to a real-time RT-PCR testing facility to get their sample tested by RT-PCR to know whether it is actually true negative or false negative. And because of its this is required because of its moderate sensitivity. So false negative and false positive, as I have said already, false negative. So it can be the negative sensitivity can be as high as 50% only. Like the negative, 50% of the patients might not get the symptom as well as maybe negative by this test, but will come positive by RT-PCR method. So if we compare the sensitivity of a PCR test with rapid antigen test, this is very less sensitive. So with PCR, we can detect even a single copy number of the viral RNA, but with this rapid antigen test, at least 10 to 1,000 molecules should be present to get the test positive. And nowadays different test devices are available, so one has to be very judgmental before using the test, whether it is molecular test or antigen test or antibody test will be used for detection of the patient. So this is the figure which shows that already I have said these are the two setup where it can be used, containment zones or hotspots, and in healthcare setting. So so this already I think I have told in earlier slides. The test can be conducted on site under strict medical supervision, and the testing should be done within 1 hour of sample collection, not beyond that, because then the false negativity will be more. Thank you. This is a short presentation, and I think this is in my opinion, because in case of high prevalence, this should be the method of choice where we can get the result within 15 minutes, and as many as possible patients can be tested with this. But if it comes negative, it should be confirmed by RT-PCR method. Thank you.
Thank you very much, madam, on this comprehensive presentation on the antigen-based test. So take-home message that we can reiterate now is that the antigen-based test, the biggest advantage is that you can do very rapid testing using a lateral flow assay, and within 30 minutes you get the results. So we can do a near patient or POC testing, and this can drive decisions with regard to the isolation of the patient and contact tracing and the institution of treatment. All this can be done in real time. But as we all know, nothing is perfect in life. These tests are associated with their own problems. And the biggest problem, as mentioned by Dr. Shanta, was its sensitivity. Unlike in an RT-PCR test, you are amplifying the target and then detecting it. So the sensitivity is taken care of. In an antigen-based test, you are collecting the sample directly. You're not doing any amplification step. It is a protein antigen. You're not amplifying. You are directly detecting it. Hence, it leads to the compromisation of the sensitivity aspect. And therefore, as per the ICMR recommendations, we are keeping this test only when there is a high pretest probability of the test being positive, meaning in a patient who is symptomatic, then a positive result is taken as a confirmatory test. However, if negative test result returns from a symptomatic patient with high pretest probability, you don't take it as a confirmatory test. You do a reflex PCR, confirm the status, and then only you finally label a patient as a positive or a negative. Thank you, ma'am, once again for your presentation. Now we come to the last presentation to actually cover all the modalities which are available. Now, I request Dr. Banerjee to please talk about the antibody-based assays which are currently
Right, sir. I will be very short and sir precise.
available.
So I'm sharing my slides. I've got just
We
five
would be
minutes.
highly obliged, sir.
Sir, just a moment. Sir, are my slides visible to you?
Yes.
Please make it in presentation mode.
Yes. Are they visible, madam?
Yes.
Okay, thank you, thank you. So I would lead forward the discussion what Dr. Shanta carried out. And as we already know that the antibodies, they are produced according, they are the protective proteins which are produced by the immune system. And here in these particular kind of test, we are detecting the antibodies. So basically speaking, there are three important antibodies what we are targeting here, but basically the commercial tests which are available, they are trying to detect the IgG and IgM. So what are these IgG and IgM? Just to briefly, this IgG is the most abundant antibody which is found in the blood plasma and accounting to 70 to 75% of the human immunoglobulin. So the important thing is that IgG, they are largely responsible for the long-term immunity after the infection or the vaccination. So one of the important thing what we are detecting in the diagnostic test is the IgG, and the second thing is the IgM, which is which accounts to around 10% in the the blood plasma, and they are the early phase immunoglobulins, and they will develop first during the acute infection. So these are the two important antibodies what we are trying to detect in the diagnostic test. Now the molecular diagnostic test already my speakers have already talked. They are the gold standards, but the problem is that at the end of the disease outcome, when the viral load ultimately becomes undetectable, therefore at this particular stage the molecular test, they will not be able to detect a prior infection. And so this is one of the limitation of the molecular test methods. And further, to add this thing, currently available the tests, they require a lot of laboratory analyzer platforms. Also there is a huge load on the short supply and the back orders from several months. So in this particular context, seeing these particular circumstances, the role of the antibody testing is very so there are two important benefits what we can derive from the antibody testing. The first benefit of the antibody testing is that it could get insight to the individuals. Like for example, if I am an individual and I want to get back to my workspace, so it can help me to find insight that whether I have that functional immunity to combat the particular COVID disease, and can I go back to. So it is giving peace of mind to me. And the second important role of the antibody testing is that it is very important for the public health officials to find the spread of the viruses in different communities, and especially because a lot of asymptomatic cases are there. So for the epidemiological surveys, and so the antibody testing would be a very crucial things. So these are the two important benefits of the antibody testing. And the principle of the antibody assays already Dr. Shanta, I will not go in the particular details. So basically it is working on the antigen and the antibody binding affinities. So all the assays which are based on these things. So basically speaking, there are two type of antibodies assays which are being done. So one is the laboratory based immunoassays, which require a reader or an analyzer to detect the reactions, and you need large laboratory setups for this thing. And the second is the cassette based systems, which can read at the point of care through a change in the color. So there are drawbacks and benefits of both the things. So laboratory based immunoassays, they can determine how much of the given antibodies, or this is called as the titer level, they can detect it in the serum level. And the benefit of these particular laboratory based immunoassays is that they have the higher sensitivity, and the specificity can be higher than the cassette based systems. And of course, the disadvantage is that, the infrastructural cost and the lot of sample processing, they are the some of the hurdles to set up a laboratory based immunoassays. And the cassette based system, already Dr. Shanta discussed with these. They are the color based assays, which you can see by the naked eyes. And the beauty of these assays is that they give the immediate results, and they are very helpful to detect the incidences on the spot. So these are the two important antibodies assays. One is the laboratory based, and the second are the immunoassays. And the function is very simple. Like, for example, when you carry out an antibody test on these particular assays, so they will detect the whether your body is producing those particular antibodies or not. So in specific, you are checking the IgG and the IgM. So this particular slide shows that, if the test sample is, showing red color, and this is also the red color, it shows that the body has produced the IgM and the IgG. So in contrast to this, if your test sample does not shows the line in front of the T, that means the body has not produced, and body has not produced IgM and the IgG. And so these are the two important interpretation what is relevant for us. So I will these are some of the guidelines which already ICMR has suggested. So my bottom take up for this particular antibodies is that in the future they will be very important for the disease surveillance and the epidemiological research, which will be very crucial in determining the course of COVID-19 in India. So the antibody testing would be very relevant in two important areas. Thank you so much, Dr. Rohit.
Take-home points from the presentation of Dr. Banerjee. The antibody tests, as you saw the surge for the RT-PCR-based tests now, with the onset of vaccination and large population of people getting vaccinated, you'll find more and more people asking for antibody-based tests.
Exactly.
There are various ways in which these tests can be used. One of them is in conjunction with RT-PCR to arrive at a diagnosis. Though currently they are not considered as a sole criteria for diagnosis, they can be used with other molecular technologies or antigen-based assays for arriving at a diagnosis, but not on their sole basis or a single test basis for antibodies.
Correct.
There has been a lot of studies available where the titers have been associated with the severity of the disease. So the measurement of titer could have a predictive value, with higher titers indicating severe disease. Community surveillance, as was enunciated by Dr. Banerjee, is a very important area. If you want to know the prevalence of infection in the community, serosurveillance is the only way you can get to know. And you might have seen so many newspaper reports coming in from Maharashtra, from Delhi, from ICMR doing its surveys all across the country. Serosurveys are being done continuously. In fact, it started in, I think, in May or June, and then we've been doing it all along to see how many of the individuals in a given population have become infected, and by this we can predict how much is the herd immunity that we've attained.
Exactly.
Finally, convalescent serum is something that everybody was asking for when we had a surge of cases as a treatment modality. It is still considered as one of the optional treatments, though we have basically a group saying that it may not be that effective. So to know whether the plasma is containing the antibodies for therapeutic purposes again can be answered. Finally, response to vaccination is another question that can be answered only by doing a serological test. That's where most of the labs will come in times to come. So thank you very much again, Dr. Banerjee, for your presentation. So what we've gathered now by discussing all these treatment modalities with regard to COVID-19 is that no one test group is perfect. And we can say that diagnostics for COVID-19 is horses for courses. So if you want to diagnose a particular infection at a particular time point, a particular assay should be used. So timing of the assays of essence value in the early part of infection when you just become symptomatic. Initial 7 days, antigen-based tests are good, but they're not sensitive. RT-PCR and other molecular assays are even more sensitive. That's why RT-PCR is considered to be the gold standard. Thereafter, the antibodies start to develop, and it takes about 15 days for detectable antibodies to be there. And by 28 days post-infection, most of us have antibodies. So thereafter, the importance of antibodies there. Uh, we talk about the metrics for evaluation like sensitivity, specificity, positive predictive value, negative predictive value. Herein, I'd like to say that there has to be a body in India which does a critical evaluation of these metrics. I think this is something which is lacking in our country, where the metrics are critically analyzed before they are launched into the market, as we have in Western world. So probably this is what we'll strengthen in times to come. I'll have a question before we take up the question from the audience. There are other sample matrices which are also being used, which are easy to collect. So, if anybody from the panelists would like to go ahead with the question, what about saliva as a sample? It has its own advantages. So where do we stand with regard to saliva as a sample? So, it's an open question. Any of the panelists can take it. Any comments on that? Dr. Jadhav, Dr. Vasuki?
I want to add a comment on this because saliva itself has a lot of antiviral properties. So when initially we worked on one of the paper we have published where we isolated salivary viruses, and we done a RT-PCR where there was one case study where there were the person was bitten for the thumbnail, and the oral hygiene was good. So we thought of isolating the virus from saliva. We found a detectable copies of virus by Cobas-Stack 48. We detected it, and the virus also we have sequenced it. So there are very few. Those are called, some people are called hyper excretors of saliva. The origin of the virus in saliva is salivary tonsillar leukocytes. So there are some leukocytes which are infected with the viruses. They may come up and give the virus, and they can spread into the saliva. And there is again, it depends on the. We found that virus in the early morning, so like early morning spit saliva. So we didn't find later. So there may be inhibitory properties may be active later once the saliva contains salivary or something like that. So the detection of COVID. The virus entering into the tonsillar leukocytes is still debatable. I don't think the detectable copies of virus can be detected through saliva, but we don't know. R&D should be done for that.
Thank you. There have been a lot of studies which have been recently carried out by in US, and they say that the sensitivity of saliva is actually comparable to that of a nasopharyngeal swab. So a lot of work is being done in US. And, I'm sure one of the kids, I was just going through the presentations of one of the panelists earlier, has developed a saliva-based antigen detection kit. And the beauty of this kit would be the ease of collection of the sample and self-collection of the sample. So there are studies which are saying that there is comparability of nasopharyngeal swab and salivary samples. Sorry, I said antigen, but it is for RNA. But further studies are required to see whether it actually becomes a reality or not. But it is really an exciting area. We got one of the questions from the expert. What is the future of RT-PCR? Let me take on that question. It's a very interesting question that has been asked. Rest assured, sir, madam, whoever has asked this question, RT-PCR is here to stay. Even if COVID goes, we can use this platform for so many other diseases, both infectious and non-infectious. Both for open system as well as the closed system, a lot of other parameters can be done. So please don't get worried about your investment. It is here to stay. Any questions from audience? We can take some more. Otherwise, we'll wind up.
No, I would like
Dr.
to.
Chawla, I actually wanted to tell you. I wanted to tell you that you have spoken. There is no system for validation of this test, but ICMR has its system, at least for COVID-19 tests. All the test kits which have been marketed by and CDSCO has approved, those are initially validated by, we have, ICMR has some validation centers in its own lab, ICMR labs, as well as some other labs, non-ICMR, where these test kits are validated and the results are shared with ICMR. And then if ICMR gives the nod, then only those tests are approved by CDSCO. So at least
Yeah, ma'am.
for, although our Prime Minister uh, is very much like Atma Nirbhar Bharat, and the indigenous kits should be promoted, but all these kits are initially, they have been validated and then only it has come to the market.
Ma'am, I perfectly agree with you. I was talking about the general system for all the kits, whether infectious markers or
General system,
non-infectious
yes.
markers. We need to have that system. We can take a cue from ICMR in this. Now that they've developed a knowledge base in carrying out the validation studies and not blindly follow the performance characteristics which are given in the kit literature. Up till now, we've been doing that only. So I think that is a platform that we not to need to create, taking the experience from the ICMR forward. Thank you very much, ma'am, for your comments. If there are no more questions from audience, I'd like to now wrap up this session. I'd like to thank all the panelists for providing their insights on the future prospects for COVID-19 diagnostics, and to the audience for putting in their queries. Finally, I'd like to thank the
Dr.
organizers for creating this platform where we could all deliberate and discuss upon this critical issue. Thank you, one and all.
Yeah, I
Over to you,
think
Madam
Dr.
Chair.
Dr.
Shivaji wanted to make a
Yeah,
quick
Yeah.
yeah,
point
Dr.
there.
Shivaji, please
So
come in.
Yeah.
see the important aspect we forget is like CB Naita say, some of the gene experts. I have concurred with some of the issues like high CT issues. So because of that, we used to get some issues with the false positivity, high CT issues, and whether anybody has encountered such a kind of issues with the high CTs and reproducibilities. And some of the lytic transport medium also, when we used, we found some issues of high CTs or false positivity. So
Yeah.
any transport medium issues or any kind of high CT issues anybody's faced till now, and any suggestions?
I think we should take it up offline, Dr.
Yeah.
Shivaji. We are actually running out of time.
Yeah, sure.
So we'll definitely take it up. We have a WhatsApp group as well. So we'll
Yes.
have the knowledge sharing there. Thanks a lot to all the panelists here who had
Thank
given
you.
their time, and different modalities of antibody testing was discussed, and it was very important as of now, as the vaccination is going on and everybody will be thinking about the antibody testing in the next in future we'll be thinking about it, and it was a very important session as such. So thanks a lot to each one of you all. I would like to give the participation certificates to all the panelists here. Deepak, can you please help us with that? Thanks a lot, Dr. Shanta Datta, Director and Scientist G, ICMR National Institute of Cholera and Enteric Diseases. Thanks a lot, Dr. Datta, for sharing your valuable inputs and your PowerPoint presentation, ma'am. And Dr. Rohit Chavla for moderating this session so well, sir, and streamlining it with a lot of good questions. And, to each panelist, thanks a lot, Dr. Rohit. Dr. V. Vasuki from Government Trivandrum Medical College. Thanks a lot, ma'am, for sharing your inputs and your, deep inside thoughts. And Dr. Shivaji from SRL Fortis Hospital. Thanks a lot, sir, for joining in and sharing your thoughts. And Dr. Saurabh Kumar Banerjee from IHMR University Jaipur. Thanks a lot, Dr. Saurabh, for giving your presentation and also writing for us in our eHealth magazine. Thanks a lot for that as well. Thank you. Thanks to each one of the panelists. We'll be bothering you again for writing to us in our magazine and also for such summits. Please be in touch with us, and we are looking for your support. Thank you. Thank you so much. Thanks.
Thank you, Madam
Thank you.
Chair. Thank you.
Thank you.